Let us now recall how these chromosomes are formed. As I have told you earlier, we have a double helical DNA, right? And this DNA then winds around which proteins? Remember, histone proteins, and they are 8 in number. So we have around 8 histone proteins like this, front and back, and this DNA coils itself around these histone proteins, and this results in coiling, then supercoiling, and then it condenses again to form a complex structure which contains histone proteins as well as DNA molecules, and then they form chromatin fibers, and then these chromatin fibers form the structures which we today call as chromosomes. So remember, we have DNA. DNA converts into nucleosome when it is associated with which proteins? Histone proteins. And when those nucleosomes coil, supercoil and condense, they form chromatin fibers. And then these chromatin fibers finally result into the formation of chromosome. So I am going to discuss the size of all these points. See, for example, we have a double-stranded, double helix DNA. It has a size of 2 nanometers, and when it coils around histone proteins, it forms nucleosomes. The size is around 11 nanometers. And then this chromatin fibers is of 30 nanometers, and then the supercoiling and finally the formation of this condensed chromatin or the chromosome results into the size 700 nanometer, and the final chromosome that we have at the end is of 1400 nanometer. So you can make out that from DNA we are talking about the formation of a chromosome. So what is a chromosome actually? Chromosome is composed of a single molecule of DNA which is associated with certain proteins, right? So chromosome is composed of a molecule of DNA which is associated with some proteins which forms nucleosomes, and after its coiling, supercoiling and condensation, finally we have chromatin fibers which form the entire chromosome. And if you see the size variation, a DNA molecule is 2 nanometer in size, and then when it forms nucleosomes, it's around 11 nanometer, and the chromatin fibers have a size range of 30 nanometers and then 300 nanometer, and when there is condensed chromatin material, it is around 700 nanometer, and when it arranges itself in the shape of a chromosome, it is around 1400 nanometer. Condensed chromosome in prophase. As I told you, during interphase, this structure, the chromosome entire structure is not visible. During prophase only, you can see these chromosome structures. So during interphase, the cell is preparing to divide, right? So after the interphase, when prophase is there in cell division, you can see these structures, that is a chromosome. So remember, DNA to nucleosome, nucleosome coils, supercoils and condenses, it forms chromatin fibers, and finally it forms chromosomes, and these chromosomes are located in the nucleus of the cell. And in case of human beings, we have around 46 chromosomes. We have how many chromosomes? We have 46 chromosomes in the nucleus of our cell. So as far now I have discussed how these chromosomes are formed and what are these chromosomes basically, and how they are linked with DNA. So remember, chromosomes, they are composed of a molecule of DNA associated with some proteins. Which proteins I am talking? I am talking about histone proteins, because these histone proteins form nucleosomes. Otherwise we also have some non-histone proteins, but that is not suitable to give you here the explanation about non-histone proteins. So I'll be just discussing about the basics of chromosomes. So nucleosomes forms chromatin fibers which finally forms chromosomes. So as you can see in this image, we have a DNA molecule which further forms nucleosomes, then chromatin loops, condensed chromatin loops, and then finally a chromosome. So this is the basis of the formation of a chromosome molecule, a unit. because chromosome is composed of DNA as well as proteins. So we have named a structure that is nucleosome, which is a specific structure that is composed of, remember I am explaining you again and again because it is important, nucleosome has 8 proteins plus DNA coils. So if I am going to explain you, it's like 4 at the front and 4 at the back, and the DNA molecules coils itself around this. So this is one nucleosome. Many nucleosomes like this arrange themselves, they coil, they supercoil and condense, and then they form a chromatin fiber. So you can see that I was talking about a single nucleosome, and then this nucleosome is coiling again and again to form a chromatin. This chromatin further condenses to form condensed chromatin, and then further it forms chromosome. This is the actual chromosome structure which you see during the prophase cycle of cell division. This structure is not visible in the interphase. So when a cell is dividing, when it reaches the stage of prophase, then this structure is visible in the nucleus of the cell. Why? Because during interphase the cell is preparing for division. Cell is preparing itself for division. So after prophase, these chromosome structures are further changed, and how this happens we have already discussed it during the cell division process. So remember again, DNA, nucleosome, it coils, supercoils and condenses, it forms chromatin fibers, and then finally a structure called chromosome is formed, which has size around 1400 nanometers. So it initially begins with from 2 nanometer, forms nucleosomes 11 nanometer, chromatin 30 nanometer, then 300, then 700, then finally the chromosome structure has a size around 1400 nanometers.